Welcome again to everybody after the coffee break. I hope you are fresh. And we continue with the talk from my colleague and friend Nicola Galvanetto, who will tell us how to relate material properties of condensates with their nanoscale dynamics.
Thank you, Milos. So today indeed I will be very happy to tell you about the work that we have been doing in Ben Schuller's group in the last few years. Not only me, but also other people in the group. And it's gonna be indeed about biomolecular condensates. So let's first try to understand what are these biomolecular condensates. This topic is, I would say, relatively popular these days. Some people likes them, some people hates them. Let's try to look at them objectively. So what you see here, it's a cell. Clearly it's an electron microscopy image of a cell, and we know since the 60s, 70s that cells are formed by compartments, things that you see here in contrast in this image, and these compartments are called organelles, and they have something in common with each other. That is, there is a membrane that separates what you have inside the compartment versus what you have outside in the cytoplasm. However, more recently, it has been found that there are some objects in the cell that look like compartments. They are as big as compartments, but and there are those green things that you see in the cells. They don't fully behave like compartments as the typical ones, because they can be formed very rapidly, they can be dissolved, and they can also fuse among each other pretty rapidly. And they are actually quite widespread in nature, in biology. What they have in common with each other is that there is actually no membrane on the surface of this, what are called biomolecular condensates, or also called membraneless organelles. So let's see how this is possible, what kind of physical laws allow them to exist. So you typically need biomolecules that are disordered, like charged. They need to interact with each other. So in this case, I'm drawing charged biomolecules. Could also be biomolecules that interact with each other via hydrophobic interactions. And if you have a relatively high concentration, we will see later what I mean by relative, but with a high concentration of these biomolecules, you observe a spontaneous phase separation of the system. Under the microscope, you're going to observe things that look like really droplets. It's called the dense phase, and then there is a dilute phase with monomers or dimers of the biomolecules of interest. While in the cell, these condensates really look like spots. In this case, there are this red, this protein that is fluorescently labeled that we see in red on the tip of this bacteria. So this is a big condensate of many proteins. So the talk of today is gonna be highlighting a specific aspect of these condensates, which I find quite remarkable. That is the fact that condensates, by nature, they work at very different scales, because they are relatively big compared to a cell. I mean, they are in the order of also microns, so they clearly contribute to subcellular organization of the cytoplasm. And therefore, there are material properties that are very relevant in this case, like viscosity, surface tension. So in order to measure these properties that determine how these condensates behave in the cytoplasm, we use assays like the one we see here in the video. So we measure the fusion time between droplets, or we directly measure the viscosity with microrheology. So these are, let's say, cellular scale level properties, more kind of also bulk properties of the material itself. But then if we zoom in a little bit more, we'll start to see that also transport properties start to matter. So diffusion of biomolecules also diffusion inside the condensates, or some biomolecules may be excluded from the condensate. So in this case, what we care about is to measure things like diffusion coefficients. So transport properties in the condensates, also exchange rates between the condensate and the dilute phase, the outside world. All right, so, but then if we zoom in even more, I would argue that a lot of action is happening actually at the molecular scale. So where we know that there is folding, assembly of macromolecules, enzymatic reactions. The problem is that at that point, at this scale, we really don't know very well what is going on, the structures, the dynamics that you have inside these condensates. Especially because even if you try to apply, for instance, cryo-electron microscopy, you see from this image it's extremely difficult to extract information because things are expected to be disordered and you cannot average out information from single particles and get a structure. So that is a problem. And so this will be the goal of this talk, to study what happens at the molecular scale. And also we will see if what happens at the molecular scale is related to this other scale, intermediate and cell scale. All right, so let's start with the specific system I've been working on. I've been working on with a human protein. It's called prothymosin alpha. This is a disordered protein. It doesn't have a fixed 3D structure. It's highly negatively charged and it is a nuclear protein. Then I've also been working with the histone H1, a more known protein. It's almost fully disordered, in this case highly positively charged, rich in lysines. And these two proteins are known to interact with each other and in the context of nucleosome in the DNA, the nucleus. And some colleagues in the lab previously found that, I found this video quite remarkable, but it's actually what's happening in the experiments too. Prothymosin alpha can sequester H1, so it competes with DNA, sequester H1 and allows DNA in this nucleosome to open. So if you mix these two proteins, prothymosin alpha H1, at low concentration, you observe these disordered binary complexes. However, if you mix them at high concentration, what you observe is biomolecular condensates, this phase separation into two phases and the formation of this nice droplet. So this will be the starting point of, this has been the starting point of my studies here. So first of all, we want to quantify some material properties like mass concentration. And we see that these condensates are clearly relatively dense, the order of 300 milligrams per milliliter, 300 milligrams of protein. But this also means that we still have the order of 70% of water volume-wise in the condensates. While in the dilute phase, the concentration is much, much lower, like 1,000 times lower. Now, then I studied the viscosity. You introduce fluorescent beads inside a big droplet, and you can track the diffusion of beads and obtain this mean square displacement curves and extract the diffusion of these beads and Stokes-Einstein the viscosity. And in this specific case, I obtain a viscosity of about 300 times higher of the viscosity of water. So this is the, what they call here cell scale properties, material properties. Then at the intermediate scale, I measure diffusion with fluorescence correlation spectroscopy. And in that case, I measure a diffusion of prothymosin alpha in the dense phase, which is about 40 times slower than what you have in the dilute phase outside of the droplets. But then, now let's focus at the molecular scale. So to measure properties at the molecular scale, I've been using single molecule FRET, Foster resonance energy transfer, which is a technique the lab is mastering for many years now. And the idea is that you need to double label your protein. In this case, we have a very short gray polymer with these two fluorophores. And the idea is that you excite, in this case with green laser light. And in principle, you should only see the emission from this so-called donor fluorophore. But thanks to phosphorescence energy transfer, there is a dipole-dipole interaction that allows a transfer of the energy, a non-radiative transfer of the energy to the acceptor, to the red fluorophore now, which is distance dependent. We can calculate the transfer efficiency counting the emitted photons, and the ratio between the red photons and the sum of the green and red is distance dependent. So we can be very sensitive between about 2 to 8 nanometers. Now here is an example of an actual experiment where we see this burst. Its burst is a single molecule floating in this chamber. So from each burst, we can really get a transfer efficiency value and plug it into this histogram that has two peaks. The one close to 0 is called donor-only peak, which is for molecules that doesn't have an acceptor. And then what we care about is the peak above 0. Tells us about, quantitatively speaking, really the distances between the fluorophores. So now, let's see the experiments on the condensate. So I have double labeled protymosine alpha, and I wanted to study the conformations in the dilute phase outside of the droplets. And there, this is a fluorescent time trace, see the bursts, and there I've obtained transfer efficiencies of about 0.55, which, considering the whole chain, is about 8 nanometers in chain dimensions. Then the question was, what happens inside these condensates? So these are experiments that required some technical, let's say, fiddling, but eventually I managed. You see that the time trace already show the bursts that are much more large, more spread. The diffusion is lower. And then here, the results. So the results for transfer efficiency shows, and these are all corrected for the different photophysical behaviors inside condensates, like slightly different refractive index. The chains are more expanded. This is something maybe we wouldn't have expected before doing the experiments, because I said before that these droplets are about 1,000 times more concentrated than the dilute phase, so chain should be pretty packed. Nevertheless, this protymosine alpha is more expanded in the dense phase. And this is because protymosine, which is a highly negatively charged protein, in the complex, in the binary complex, in the dilute phase, is basically wrapped around a single H1. So they dance together, they are dynamic together. While in the dense phase, protymosine alpha can share its contacts with many other potential partners. And so that's our rationale for this. We'll see more about it later. So then I was actually more interested about the dynamics more than the conformations. And for dynamics, here I really mean the dynamics of biomolecules, of intrinsically disordered proteins. This is something that maybe if you are more familiar with global, let's say folded proteins, this is something of less interest, because there you really care about the position of single atoms and to reconstruct the structure. But in this case, these proteins are disordered, so you can, generally speaking, have two major quantities to describe them. One is the expansion of the chain, what we saw in the previous slide, and the other thing is how rapidly this chain, this protein, explores space. Another way to see it is to say, how long does it take for this protein to forget about its previous conformation? Okay, so this is what we call reconfiguration time, qualitatively speaking, but in practice, what we are really looking at is the time it takes for this chain to go from an expanded conformation to a compact conformation. We basically do the autocorrelation of this signal. So clearly we don't have this die-die distance exactly, this is from a simulation, but we measure photons from expanded conformations and from compact conformations. So the photon arrivals change in relative ratio between green and red photons. And so we can correlate them and obtain some plots that look like these. These are called, that's what we call nanosecond FCS. This is a cross correlation between donor and acceptor photons, and we see this, let's say, relaxation time. And this is really the relaxation time of these fluctuations. So we fit it with an exponential, and this is the value, this reconfiguration time. So then the question was, what about the dynamics in the dilute and dense phases? So what would we expect? So in the dilute phase, that's the data I just showed you, 126 nanoseconds. In the dense phase, to our surprise, I only measured 380 nanoseconds. 380 nanoseconds is only a factor of 3 slower than what you have in the dilute phase. So let me recapitulate the quantities we showed before. So viscosity, about 300 times higher in the dense phase compared to the dilute. The diffusion in the dense phase is about 40 times lower, while at the nanoscale, the chain dynamics is only a factor of 3 slower. So at this point, we were a bit puzzled, so we want to understand more. In order to understand more, I asked my colleague Milos and Robert Best in US. They did all atom molecular dynamics simulation. These are actually pretty heavy simulations. 4 million atoms, 200 proteins, 6 microseconds. They ran in a Swiss supercomputer for several months. And of course, first we made sure that the observables of the experimental observables kind of matched the prediction from the simulation to see if they were close to reality. And then we started to look at things that we cannot look experimentally, like contact dynamics. So here in blue, you mostly see lysines, it's the H1 lysines, and in red, mostly glutamic acid and aspartic acid from protymosine alpha. So negatively charged residue and positively charged residue form these contacts, and we made some statistics of these contacts, finding that the average contact lifetime of these single residues is about 2 nanoseconds, which is much, much faster than the overall chain dynamics, 380 nanoseconds. What does it mean? This already starts to tell us that a single chain is clearly moving in a sticky environment. But this sticky environment is sort of continuous. It's more like a continuum of stickiness. And this we'll see in a while I'm highlighting this aspect of a single chain in a continuum environment. So, all right, so now we have a clearer picture of what is going on in the cell, in these condensates at different scales. And my, let's say, actually original motivation to start working on this project was, but would it be possible to link these quantities, things like chain dynamics to diffusion coefficient to viscosity? Because intuitively speaking, you may say, sure, if you have a chain that is reconfiguring very slow, it's more likely that you're gonna have a pretty high viscosity, but is there maybe some mathematical relations, some models that can describe this, these different scales? So to answer this question, with my student Simone Del Grosso, we expanded the data set, started using also protamine. Protamine is also protein that is related to DNA. This is a short disordered proteins rich in arginine compared to H1, which is rich in lysine. And protamine is an interesting protein because it basically replaces the histones to compact DNA even more in sperm cells. So this phenomenon is, by the way, not very well understood or known yet. And then we also studied polylysine 50 repeats of lysine and polyarginine 50 to, let's say, specifically focus on the different nature of the positively charged residues. So we start mixing these, and they form droplets that are proteinose alpha plus these positively charged biomolecules. They form droplets that are qualitatively very similar among each other. But then we started measuring chain dynamics. So this is the H1, the one I already showed you, 380 nanosecond reconfiguration time, which correspond to, say, this dynamics inside the condensates, if you could really see it. But then I measure polyarginine 50 proteinose alpha, and look at this. So this is 13,000 nanosecond reconfiguration time, meaning it's about 50 times slower. And in fact, this is actually a simulation for protamine, but you see it's very similar reconfiguration time. You see how much slower the chain dynamic is. Of course, the time scale is the same. Let's say the frame rate is the same. So, so we measure all these samples at different salt conditions. And we can see now, without going too much details, that a general trend is for each sample, if you increase the salt in the buffer, chains becomes more dynamic. Clearly this is a charge-charge interaction, so you sort of grease it with salt. But also arginine-rich condensates tend to be much slower, higher reconfiguration time, so means slower compared to H1 or polylysine 50 that are lysine-rich. So there is this big gap between these condensates. So this is the molecular scale, but then we measure with Simone the diffusion coefficient, diffusion times, and look at the trend, very similar trend. The pink is up, so arginine 50 proteinose alpha is on top, and then H1 at the bottom. And then we also measure viscosity. So this is, I would say, the bulk viscosity with beads. And the viscosity shows the same, similar trend. So what would you do? I clearly plot one against each other in a 3D logarithmic plot. And we see that this cloud of points really align, but in particular, the slope is 1, meaning that these are linear relationships, which is, let's say, maybe intuitive, but clearly something I would have necessarily bet on before. So there is linear relations between these scales. So I wanted to find models to see if I could interpret this quantitatively speaking. So I started to use polymer models, classical polymer models. So here we have our data in diffusion coefficient versus viscosity. And I first started applying the Rouse model, arguably the first polymer model ever studied or published, at least. So this was developed in 1953, so quite many years ago. And you really just consider beads and springs. So basically what you can vary is the friction applied to beads. and that's it, and the number of beats. So this model would predict a viscosity from a diffusion coefficient that is actually a very simple relation, and that's this estimate, which already when I started plotting it, I was quite astonished because, I mean, this is a very simple relation. This is not a fit clearly. These are all quantities that you can plug in. And they are wrong by less than an order of magnitude, but like a factor of 4. So quite well done with the Rouse model. But then, let's say another iteration on top of the Rouse model has been the Rouse model with entanglement proposed by Gilles Dejean in 1971. And in this case, you have to consider that polymers are not completely free. They cannot overlap with each other, and they have sort of tube that describe the space where they can move inside the mesh, inside the dense phase. So in this case, the viscosity is also very similarly predicted, except that there is another parameter that has to do with the ratio between the size of the protein and the tube diameter. And in this case, the prediction is very remarkable. And the predicted tube diameter is what we expect in terms of mesh size that we measure, is the order of 3 nanometers, 3-4 nanometers. And I think this is quite amazing, especially because these are not only my points, but you see here that there is a black and a green point, and those are data points that I didn't collect. I found from the literature for LAF1 and A1 LCD. So I was in a way very happy and surprised for this. These relations, by the way, also work in terms of diffusion coefficient and reconfiguration time, and reconfiguration time versus viscosity. So the model really seems to capture, let's say, the physical nature of this of these interactions. However, what I cannot capture here is that if you just give me a sequence, I cannot place you the point in this plot. However, if you measure just one of these quantities, it really looks like the other two are kind of predetermined. So let me conclude then, summarizing that in fact, there are relations between quantities at different scales, like viscosity, diffusion coefficient, and reconfiguration time, thanks to the Rouse model with entanglement. And let me just finish saying what do this finding are really teaching us more in terms of maybe biophysics and also in terms of more biology. So one aspect is that I was going through the talk by comparing this 300 times more viscous in the condensate and then just saying it's just a factor of 3 slower chain dynamics in the condensates. So the models that I showed you actually highlight the fact that the problem is not what happens in the dense phase. In the dense phase, things are pretty much described by the Rouse model with entanglement in this case. The sort of anomaly, if you want to call it this way, is what happened in the binary complex in the dilute phase, which despite being almost in water, so 1 proteinose alpha, 1 H1, they reconfigure very slowly. So the interactions are relatively strong. So that's somehow what is weird in that regard and what may be worth exploring further. In terms of biology, when I mentioned the electron microscopy or tomography on condensates, I didn't actually say completely the truth, because there are some recent studies that attempt to study actual chromatin phase separation, or even in the cells, where you can really recognize single nucleosomes in the condensate, because those are big enough and you can also average them out and obtain a structure. And in this case, here is where I really see that matters the relation between the nanoscale dynamics of chains, which is what really matters for instance, DNA dynamics, transcription there, translation there, and what happens more at the mesoscale in the nucleus, where these domains of chromatin can fuse, can change shape, so a more large-scale remodeling of chromatin. So I think what I've shown is that there is actually really a link between these two scales, and so we can sort of predict one dynamic by looking at, let's say, for instance, viscosity, which is much more easier to measure. So with that, let me thank you for your attention, my colleagues, Milos, Ben Schuler, head of the lab, Robert, and all the people in the lab, and I'm very happy to take questions.